In part B of our series on the molecules of life, we'll talk about the four major types of macromolecules that make up the bulk of a cell. These are carbohydrates, also known as polysaccharides, nucleic acids, lipids, and proteins. Let's start off looking at the chemical composition of various types of cells. First, we can look at an E. coli cell and look at what percent of the total mass of an E. coli cell is each type of molecule. So in an E. coli cell, like in many types of cells, the bulk of it is water. But if we dry up an E. coli cell, maybe gently dry it and have only the powder, the dried powder left from an E. coli cell, what we'll then get are a bunch of molecules, and what we see is that, and these are color-coded, about half of it's protein, here shown in orange, and then we have RNA in yellow, DNA in green, lipids in blue, and polysaccharides in green. All of these are what we call macromolecules. Macro for large. And then we have some small organic molecules, and we have some inorganic residue as well. So what we see is that when we go to human cells or plant cells, again, the bulk of the cell mass are macromolecules. What differs between the types of cells is the relative proportions of the macromolecules. For instance, human cells have much more lipids than other types of cells, and plant cells have a lot more polysaccharide than other types of cells. So these are the questions that I'd like you to think about and focus on in this unit. What are the major types of macromolecules? And this is something that you really need to know. And what are they made from? All macromolecules are assembled from smaller subunits, and so you need to know what the monomer or subunit and macromolecule polymer relationship is. Okay? So the function of these macromolecules depends very much on their structure. Okay? So understanding what causes these different macromolecules to assume the exact precise shape that they do required for the function is important. And finally, we talked about how evolution is a change in the gene pool of a population, and that natural selection acts on genetic variation. Well, how does it do that? Genetic variation causes variation in either the structure of a macromolecule or the composition of a macromolecular assemblage. Okay? And these changes in the structure or composition affect how these macromolecules work, and how ultimately how the organism interacts with its environment and will make a difference in the survival and reproduction of the organism. So evolution then really depends on genetic variation. Genetic variation works through variation in structure and composition. For more on the learning goals associated with biological molecules, I urge you to visit my blog. I have a blog post on biomolecules, and you can follow that link. So let's start off with carbohydrates. Carbohydrates are important, of course, for energy metabolism. They store energy, and glucose is the main molecule used by cells as fuel to power the work that they do. So they store energy in the form of polysaccharides, these large chains of carbohydrates. And Other molecules, in addition to storage, polysaccharides can have structural roles. For instance, cellulose fibers make up the bulk of the cell wall for plants. So in carbohydrates, the name implies that you take a carbon plus a water molecule, and when you combine them, you get the formula for a carbohydrate. Okay? So if you take the CH2O and repeat it n number of times, you get the general structure of or the general composition of a carbohydrate. The monosaccharide glucose, which is also a hexose, meaning it has 6 carbons. So it has 6 carbons, 12 hydrogens, and 6 oxygens. It's basically this CH2O multiplied by 6. Okay? So as I said, glucose is a monosaccharide. These monosaccharides can be linked together through what are called glycosidic linkages. Glycosidic linkages or glycosidic bonds. And when you have 2 sugars linked together, then you have a disaccharide. And of course, you can have longer chains and get polysaccharides. Now, because of the glycosidic linkages and how they're formed, you can have branched glycosidic linkages. For instance, you could have a glycosidic linkage here and another one here, resulting in a branched chain. Next, we'll look at nucleic acids, DNA and RNA. And we all know that they're used for storage of information, basically genetic information. And particularly for RNA, processing genetic information in the cell. So that's their function. Now, when we look at their structure, what we find is that they have a rather complex structure. Okay? Their monomeric subunits are nucleotides. And this figure shows you what a nucleotide is made of. First of all, it has a 5-carbon sugar. Okay? Ribose, a pentose, means it's a 5-carbon sugar. The 5-carbon sugar in RNA is called ribose. The 5-carbon sugar in DNA is called deoxyribose. The only difference is that the hydroxyl group, the OH in ribose, is substituted with just an H, a hydrogen. So that's the deoxy, meaning this oxygen is missing in deoxyribose. So deoxyribose, of course, is the sugar for DNA, and ribose is the sugar for RNA. Okay? That's the one key difference between DNA and RNA molecules. We won't talk about nucleosides very much at all. We will talk a lot about nucleotides. So nucleotides means that they have phosphates attached to them, between 1 and 3 phosphates. So in addition to the sugar and the phosphate, the ribose is attached to a nitrogenous base. Okay? So purines have 2 rings, and pyrimidines have 1 ring each. So these are the nitrogenous bases, have nitrogens in them. Okay? And I think these R groups refer to the either the ribose or deoxyribose. This is how they're attached to ribose or deoxyribose. Okay? And of course, thymine is in DNA and uracil is in RNA. They both have adenine, guanine, and cytosine. And so how are these linked together? These nucleotides, remember they have to have a phosphate, are linked together by phosphodiester bonds. And here you go. Now here's a nice chart, and I think a good way for you to organize the information about macromolecules. We'll be discussing proteins next, so you can finish filling in this chart, but you should have enough information now to fill in the information for nucleic acids and glycans. Now on to proteins.